Hi, my name is Kobir.
Hi, I'm Adriana.
Hi, I'm Fahmi. We are from Group 4. Will present the EEG, open and close analysis. For that, we need to know first what is EEG. EEG is a machine that measures electrical activity in the brain, which will evaluate subjects' brains' electrical impulses and send signals to a computer, which in an EEG recording, the electrical impulses appear as wavy lines with peaks and valleys. This thing we will be discussed in the discussion. There are four basic brainwaves with their own frequency, which is delta below 4 Hz, between 4 Hz to 8 Hz is theta, 8 to 13 is alpha, and 13 to 30 is beta. In our data analysis, there is also motion artifacts. So what is motion artifacts actually? Motion artifacts is an unwanted electrical input caused by physical motions of the body and measurement instrument, which divided into two major parts, which is the first is the motions of the measurement system and second is motions of subjects generate electrophysiological signals, EOG, electrooculogram, and EMG, electromyogram. In our case, we had the muscular artifacts, which is the EMG in our, as you can see, at the right side of the slides. And the left side shown the types of physiological signal artifacts.
Hi, my name is Kobir and I will be presenting the methodology and the result of our report. Firstly, for the methodology, our first step is to handle the acquisitions of the EEG data, which in this case we use MATLAB to perform further analysis on the raw EEG data for both the eyes closed and open situations. Secondly, we proceed to the data preprocessing. For the data preprocessing, we plot and label the raw data for the EEG signal and for each electrode, which are 63, 64, CO1, and CO2. For sample rate determinations, rates of EEG equipment should be anywhere from 120 and until 512 Hz, and 256 Hz is the frequency we choose to study in this report. Signal is converted into the frequency domain with an example being S2 equal to spectral, bracket S1, comma segment size, comma shift or step size, in which the shift size is equal to half of the segment size. For the bandpass filter, the bandpass filter with the frequency of interest ranging between 8 and 13 Hz is the most suitable for filtering the signal. This is because the alpha wave is the most prominent type of brainwave, and it is the one that is most likely to be picked up in the data sets of the resting EEG. Pictures on the right side show the information needed to be used to compute the bandpass filter for the filtering of the data. W is equal to frequency of interest rate divided by the half of sampling rate. The bandpass ages frequency WP used is 8 to 13 for the frequency divided by half of the sampling rate, which is the sampling rate is 256 Hz. For stop band ages frequency WS used is 6 to 15 Hz for the frequency divided by half of the sampling rate, which is the sampling rate is 256 Hz. Passband ripple RP and stop band ripple RS used are 1 dB and 10 dB. Lastly, for the data comparison, the unfiltered and filtered signals in the time domain as well as the frequency domains are shown. The filtered signals for eyes closed and eyes open are being compared with each other. Next, these are the MATLAB algorithm we use to pre-process and interpret the data. The first one is eye closed. For the raw signal, we need to convert the table into double form, which is S1 equal to table 2 rate bracket eye closed, and then plot the raw signal, which is S1. Next, for the bandpass filter, we use the information mentioned on the previous slide to define the bandpass filter. Two functions that are used for the bandpass filter, which are filter FS1 and spectral FS2. The reason why we need to filter the data is to remove the mean and also to remove the low frequency of the movement artifacts. Next is the MATLAB algorithm for the raw signal of EEG electrode. First, we define intervariable for each electrode and then we plot the graph using the codes shown below, similar to the MATLAB algorithm for unfiltered versus filtered signal. We define the filtered signal for each electrode and use the MATLAB code shown below to plot the graph. for plotting raw signal S1 in frequency domain, which we label it as S2. We use the algorithm below to plot the signal in frequency domain. Same goes for the eyes open. We define the raw signal as the S3 and S4 for the raw signal in frequency domain. Lastly, we compare the eyes closed and eyes open using the MATLAB algorithm below. Next, moving on to the result obtained from interpreting the data using the MATLAB algorithm mentioned previously. For eyes closed, these are the shown for the raw signal S1, and below is the graph for raw signal of the electrodes. Graph on the right side showing the S1 in frequency domain, which we name it as S2. From the plotted graph S2, we can see that the electrode C3 shown the highest peak amplitude. Next, we have the unfiltered S1 signal versus filtered S1 signal, unfiltered S1 signal S1 versus filtered S1 signal FS1 in frequency domain, and unfiltered versus filtered signal for each electrode. For the filtered signal FS1 in frequency domain, we can observe that the graph shift and the highest peak amplitude is now on electrode O1. Next, same goes for the eyes open. We have raw signal S3, raw signal for the electrodes, and the S3 signal in frequency domain, which we name it as S4. For the S4, we observe that the electrode C4 shown the highest peak amplitude. Next, we have the plotted graph for unfiltered S3 and also for filtered S3. Unfiltered, and we also have the unfiltered S4 signal versus the filtered S4 signal, and unfiltered versus filtered signal for each electrode, which all of these lies under the eye open data. Lastly, these are the comparison between filtered eyes closed and filtered eyes open in the frequency domain. That's all from me. I shall pass to Adriana to discuss further on the result.
Hi, I'm Adriana. So I'm going to proceed with the discussion part. So in our study, we utilized the fast Fourier transform FFT as the method to extract brain signals, primarily the alpha waves, from the EEG signals using the MATLAB software. So this method employs mathematical means or tools to the EEG data analysis. As for to analyze the waveform, spectral analysis or the frequency domain analysis is employed as the signal processing technique. So to be specific, FFT for spectral analysis of EEG signals in the frequency domain is used for alpha wave detection in the eyes closed and eyes open state in this paper. As mentioned in the introduction part, each of the 5 primary EEG signals has their own range of frequency, in which they are associated with varying level of arousal of the cerebral cortex as shown in the table. So it is noted here that the level of cortical arousal decreases as the frequency of the EEG patterns gets lower. So based on the state of EEG rhythms associated to, it is expected that alpha waves is prominent during the eyes closed state and less prominent during the eyes open state, and that is our hypothesis. So after the pre-processing of the signals and feature extraction, the filtered signal for the eyes closed is compared to the filtered signals for the eyes open, particularly at the occipital regions, which corresponds to the channel O1 and O2. So upon our findings, it is observed that the amplitude of the alpha waves during the eyes closed condition is significantly higher than in the eyes open condition, as shown in the figure here. So does this satisfy our previous hypothesis? So, the result is also coherent to the findings of the previous study where the alpha band activity in visual brain regions increased when people close their eyes. So this was first identified by the researchers who subjected their participants to light, and changes are observed in their occipital alpha activity. So the reduction of the alpha waves in the eyes open state is also well described by a phenomenon called alpha blocking, or previously known as the Berger's effect, in conjunction to Hans Berger discovery of human EEG studies. So in brief, the alpha blocking refers to the reduction in the spontaneously recorded alpha band power at the occipital region in response to the opening of the eyes as a transition from the eyes closed condition. So last but not least, in reference to a recent study, we found this paper which conducts a study on EEG signals during the eyes open and eyes closed condition. So the purpose of this study is to identify the significance of the recorded EEG signals during the two mentioned states. For the method, the study utilizes the discrete wavelet transform method of analysis, to extract the EEG signals, and then further interpretation is done in terms of the sideband frequencies, which is represented by the coefficients. So the average percentage of the wavelet energy is then plotted against the wavelet coefficient for both the eyes closed and eyes open condition, as shown in the graph below. So from the plotting, it can be deduced that when a person closes his eyes, there will be a significant increment in the alpha waves, a reduction in both delta and theta waves, while the beta and gamma waves will remain stagnant.
In this part, I will show you the suggestions to remove the motion artifacts. Actually, before this, in the methodology and results part, Kobe had showed to us how to remove the motion artifacts in our data analysis. But for my part, I will give the other suggestions to remove it. So from the left side of the slides, you can see the recent advances in artifact removal technique. But the most well-known technique for artifact suppression is the BSS, blind source separation, which consists of two types, which is ICA and CCA. ICA is independent component analysis, and the CCA is canonical correlation analysis. Artifacts components are manually or automatically selected and then deleted from the separated components, resulting in a clean EEG. The other types of suggestions is the automatic artifact removal AAR toolbox V1 version 1.3, which is for MATLAB. So there are three-step general purpose artifact removal technique, which is the first, the EEG data is divided into many special components in the BSS. Second, suitable criterion is employed to recognize artifact related components automatically, which is to choose whether to remove EOG or both EOG and EMG using the spatial filters. And last is the only non-artifactual components are used to recreate the EEG data.